Genetic diversity drives evolution. Evolution allows organisms to accumulate changes over time that help them survive better in their environment. Sex and its ability to shuffle chromosomes and genes to create new combinations and new organisms is a major driver of evolution in eukaryotes. But what about prokaryotes? Phenomena like binary fission are asexual. How do prokaryotes achieve genetic diversity? How do they evolve and adapt to survive their environments? I'll answer that question, or those questions, and more in this video. By the end of this video, you should be able to define the term horizontal, aka lateral gene transfer. You should be able to explain DNA recombination. And by explain, I mean in a sentence or two. I'm not looking for you to be able to go into the details of how it happens. You should be able to explain two ways that prokaryotes acquire new genes and increase their genetic diversity. And then you should be able to explain how horizontal gene transfer can lead to antibiotic resistance. Now, this is part one of 2 videos on this topic. So you may find that you need to get through all of this lecture, both parts, before you can accomplish all of these objectives. All living organisms, both prokaryotic and eukaryotic, use DNA as the blueprint for synthesizing proteins and RNAs that are essential for life. The segments of DNA that encode these proteins and RNA are called genes. But what happens to the DNA and those genes in a cell? Well, there are 3 things that are going to happen. The first is that the information from the DNA or the genes can be transcribed. That is, it can be copied into RNA, and then that RNA gets translated to produce proteins. Collectively, we call this process gene expression. So one thing that can happen to the genes and DNA in a cell is that they get expressed. Another thing that can happen, this is number 2, is that genetic information can be replicated. Okay, so it can be replicated. So we know that all cells copy their chromosomes, and then they pass those chromosomes on to offspring, which we call daughter cells. So information can go from parent to daughter cells or offspring. Another thing that can happen is that genetic information can be exchanged between cells that are in the same generation. This is through a process called recombination. So there are 3 things that can happen to the DNA in a cell. It gets expressed, it gets replicated and passed on to daughter cells, or it can be recombined, transferred within cells in the same generation, and then recombination. So when DNA is replicated and it's passed on to offspring cells, aka daughter cells, this is called vertical gene transfer because the DNA is getting passed from one generation, which is the parent generation, to another generation, which is the offspring generation. It's kind of like when people have children, they pass on a copy of their DNA, or a copy of their chromosomes, onto their children. Whether or not their genes get expressed is in the domain of gene expression, but the fact remains that DNA was passed from one generation, the parent, to the offspring. And all cells do this, prokaryotic and eukaryotic. Prokaryotes are capable of another type of gene transfer, which is called horizontal gene transfer, or sometimes it's called lateral gene transfer. Here, genes are transferred from one cell to another, but in the same generation. And this is going to involve the process of recombination. It's kind of like if you, let's say, passed on the genes for your hair color and hair texture to a friend, or maybe somebody you met while you were out grocery shopping or at a barbecue or at the beach. They are in your same generation, and you would be passing those genes on to them. Can you imagine how weird that would be? Like you're out minding your own business, going about your day. Somebody taps you on the back because maybe you dropped something, and then suddenly you're a redhead or a blonde. That's kind of what horizontal gene transfer is. Now, for the remainder of this video, we'll describe horizontal gene transfer in bacteria. But it's important for you to remember that for each of the methods that we'll talk about, there's going to be the exchange of genes between two different DNA molecules. One molecule will be the bacterial chromosome, and the other will be a bit of foreign DNA. The foreign DNA may be absorbed from the environment, or it could be donated by another bacterium, or it could be donated by a virus. Whatever it is, the result is the same. We're going to get two DNAs that become one because they share similar but not identical DNA sequences. And then there's going to be proteins that direct the exchange of DNA. There are three types of recombination. You have general recombination, like what we see with eukaryotes during meiosis, where you have the reciprocal exchange of DNA sequences, okay, between two different DNAs. Now, in addition to meiosis and crossing over, we are going to see this in prokaryotes during a process called transformation and another process called transduction. A second type of recombination that we see in prokaryotes is called site-specific. Here we're going to have a DNA, a viral DNA specifically, that gets incorporated into a bacterial chromosome. So the process where we'll see site-specific recombination is called transduction. A third type of recombination is called replicative. Here we have genetic elements that are going to switch from one position to another within the chromosome. And this is through a special process called transposition. I've put a video in our lecture videos folder that describes in some detail how general recombination happens. Now would be a good time to watch that video and rejoin this lecture when you're done. The first type of horizontal gene transfer that we'll look at is called genetic transformation. You'll want to write this down. Genetic transformation is the transfer of DNA from one cell to another as naked DNA. Most bacteria are surrounded by other bacterial cells, either of the same species or of a different species. As happens with any living thing, occasionally some of these cells die. When dead cells begin to decompose, very often enzymes in the cell begin to break down molecules like DNA, creating fragments. And then those fragments get released into the environment. That's the naked DNA. Many bacteria have the ability to absorb those naked DNA fragments and incorporate them into their own bacterial chromosome as a mechanism for repairing broken genes or increasing genetic diversity. These cells that can absorb DNA from their environment are called competent cells, and they're able to make holes in their cell wall that allow foreign DNA to enter. Only recently have we observed that these cells actually make pili, and they use those pili to lasso the DNA in their environment and bring it back to the cell. I put a video showing how the cells do this in your lecture videos folder, and I highly recommend watching it. It is so cool. When naked DNA fragments have chi sites in them, recombination is likely to occur. At this site, the DNA is nicked, that means it's cut, and a protein is added to the floppy DNA end, like here. This protein is then able to probe the nucleotide sequence of the bacterial chromosome, looking for complementary sequences. When one is found, the DNA actually displaces one strand of the chromosomal DNA, and the enzymes that are bound to the DNA will continue to open the chromosome and match sequences together. So this continues to happen right here. So what I hope you'll notice is this fragment has genes A, B, C, and D, and that's one form of each of those genes. The chromosome here has the same genes, just a different form. So what's gradually happening is we're replacing the chromosome version of the gene with the gene that's coming from the environment here. Eventually, the foreign DNA strand gets chewed up and the foreign DNA gets incorporated into the chromosome. So again, the video on general recombination kind of describes this. Okay, so this is what happens during transformation. We have a fragment of naked DNA that gets partially incorporated into the bacterial chromosome. So the first widely accepted demonstration of bacterial transformation was reported in 1928 by British bacteriologist Frederick Griffith. Griffith was interested in the epidemiology and pathology of Streptococcus pneumoniae, a common cause of bacterial pneumonia at his time. Now, Griffith knew that there were at least two different types of S. pneumoniae. One type, which he designated R, made rough, dry-looking colonies when it was plated out on a petri dish, and this particular type was not known to cause illness in humans or other animals. The second type of cell was called the S-type. S-type bacteria made copious amounts of glycocalyx, so much so that instead of getting a nice circular colony, you got these kind of blobby, snotty looking colonies, like what you see in this picture here. This particular type was deadly. Remember, this is before antibiotics. Griffith is doing his experiment around the same time that Alexander Fleming is going to first discover the penicillium molds. And this is at least 10 years before Florey and Chain isolate the penicillin antibiotic and figure out how to make it in large enough amounts to turn it into a therapeutic. So at Griffith's time, pneumonia could be deadly. So here's the experiment that he did. He took S-type Streptococcus pneumoniae. These are the ones that make the mucousy colonies. And he injected that bacterium into healthy laboratory mice. Then he waited to see what would happen. The mice got sick and died. So then he took samples of the various tissues and fluids in the mouse and tried to isolate bacteria. And he was actually able to isolate the S-type Streptococcus pneumoniae bacteria from the mouse on a petri dish. The next thing he did was he took the R-type. Remember, this is the harmless type of bacterium. He injected that into a mouse, waited to see what would happen. The mouse stayed healthy, didn't die. So then he took samples of the mouse's tissues and blood, streaked that out on a petri dish, and he got back the R-type colonies. So then he decided to take the S-type, that's the deadly bacterium. He heat killed the bacteria and then injected the heat-killed bacteria into a healthy mouse. Well, as you might expect, the mouse survived. At this time, Griffith wasn't sure whether it was something that the bacteria were producing that was leading to death, or whether it was some component of the bacterium itself that was causing death. So if he used heat-killed cells and it was something in the bacterium itself that was causing death, then these heat-killed bacteria should have also led to death, but they didn't. So the mouse lived. He tried to recover the S. pneumoniae bacterium from the mouse. He was unable to do it. Next, what he did, and this is the most interesting part of the experiment, he took the material from the heat-killed experiment, So he took some of those heat-killed S. pneumoniae and he mixed them with the R-type S. pneumoniae that were alive. Now, in theory, neither one of these two materials, the S-type living cells and the heat-killed S-type, neither one of them led to the death of a mouse previously. But when he mixed them together and injected them into a mouse, suddenly the mouse died. And even more interesting, when he tried to isolate the bacterium from the mouse, instead of getting the R-type cells, he got S-type, suggesting that some molecule in those S-type bacteria was transmissible to the R-type, and it could convert the R-type into S-type. This is before anybody knew about DNA. Remember, the DNA experiments came in the 1940s. So there's still a huge question over these experiments. What was it exactly that was transferred? But the experiment shows definitively that something can be transferred and that it changes the cells. That's why this is called transformation. So if one of those naked DNA fragments had a gene for antibiotic resistance on it, then that means that the cell that picks up that DNA and is able to incorporate it into its chromosome by recombination could also pick up antibiotic resistance. Another way that bacterial cells transfer DNA is through conjugation. Conjugation is the closest that bacteria get to sex, but it's really not sex. It's really more like snuggling. So there's a special type of plasmid called an F plasmid. The F stands for fertility factor. And cells that have this F plasmid are able to make the structures necessary to transfer the F plasmid to other cells. So a lot of your gram-negative cells that do this, that have the fertility factor, they'll make something called a sex pilus. So remember back to chapter 4 when we talked about pili, they are these kind of tube-like or straw-like structures that get made when bacteria assemble pillin proteins one on top of another to make a long chain. They can touch a surface and then they can break apart the pilus and bring themselves towards that surface. This was like the grappling hook motility that we talked about, right? Well, it turns out that cells that have these F plasmids, they're able to make a special pilus, that's the sex pilus, and they can transfer DNA through that sex pilus to another cell. Cells that don't have a fertility factor are called F minus cells. So in this picture, you can see an F positive cell that's making these fimbriae here, okay, and a sex pilus, and it's going to transfer the fertility factor that it has to this F minus cell over here on the side. Gram-positive cells can do the same thing. Many of them will actually make a sticky, like an extra sticky coating on their surface, and as they tumble in space, they'll stick to other cells through this coating, and then they'll make something called a mating bridge. And fertility factors can be transferred from one cell to another via the mating bridge. So it's the same thing. Okay, this slide is just here to remind you about what plasmids are, little extra chromosomal circular bits of DNA. There are many different types of plasmids. You have what's called a conjugative plasmid, so it carries the genes for making the sex pilus or the mating bridge, and it has genes that are going to help transfer a copy of the F plasmid from one cell to another. Another type of plasmid is called a dissimilation plasmid. So it encodes enzymes for breaking down unusual compounds like octane and your catechols, which are compounds that you find in pesticides and fragrances. These dissimilation plasmids allow some bacteria to actually break down adhesives. So in a hospital environment, there's a bacterium called Pseudomonas aeruginosa, which is transferred in the hospital. It's what we call a healthcare associated infection. In any case, it can get under the caps of bottles and actually live under the seals in a bottle of sterile saline or some other solution, thus contaminating that bottle and making it more likely for the bacteria to be transferred to a susceptible patient. These Pseudomonas aeruginosa also have dissimilation plasmids that allow them to break down common disinfectants. R factors are another type of plasmid. They encode genes for antibiotic resistance, and they can have up to 8, they can confer resistance to up to 8 different antibiotics. Now, your dissimilation plasmids and your R factors can also have the genes for conjugation, so they can also be technically considered conjugative plasmids as well. So here's an example of an R factor. These are the genes over here for making a pilus and transferring a copy of this plasmid to a new cell. Over here is the origin of replication. So the plasmid can actually reproduce itself inside of a bacterial cell. So you have multiple copies of it. And then these are the antibiotics that this plasmid gives resistance to. So a cell that gets this plasmid would be resistant, for example, to tetracycline, to streptomycin, to sulfonamide, all sorts of things. I'm sorry, not sulfonamide, sulfamethoxazole. Here's how conjugation happens. We're going to start out with our F positive cell. Remember, this is the one that has the fertility factor plasmid in it. Because of gene expression, the cell will start to make a conjugation pilus or that mating bridge. That's what this is right here. The plasmid will be nicked by a protein, so that means that one strand of the plasmid will actually be cut. And then proteins from the conjugation plasmid will bind that nicked DNA and transport it across the conjugation junction, that mating bridge, or the conjugation pilus, the sex pilus. This will deliver one copy of the plasmid to an F- cell. Then what happens is we'll have a single-stranded fertility factor in this cell, and then a single-stranded fertility factor over here too, because one strand was donated to this new cell. The DNA replication proteins inside of the cell will then bind this single-stranded F plasmid and synthesize the second strand. So then we'll end up with double-stranded DNA plasmids in both cells. If your head is spinning at this point, you don't need to know this in so much detail. You need to know that conjugation, and you'll probably want to write this down, conjugation is the transfer of DNA from one cell to another via a sex pilus or mating bridge. That's what you need to know. If the plasmid has genes for antibiotic resistance on it, then that means that the F- cell will pick up genes for antibiotic resistance. So again, those R factor plasmids and dissimilation plasmids may also be conjugative plasmids because they have the genes for transfer of the plasmid. Sometimes the fertility factor plasmid can be incorporated into the bacterial chromosome via recombination. So we get what's called an HFR cell, or high frequency of recombination cell. And what's important to know here is that when this recombination happens, any genes that are part of the plasmid, they become part of the chromosome now. Even though the fertility factor is now incorporated into the bacterial chromosome in this HFR cell, you can still have transfer of DNA. It's just that part of the bacterial chromosome gets transferred with the fertility factor into the new cell. And recombination can happen between this new bit of chromosomal DNA from our F plus cell and the chromosome of the F minus cell. Notice that because the fertility factor didn't come with that DNA, this cell that received the DNA is not called an F plus cell now, it's called an F minus cell. Horizontal gene transfer, also called lateral gene transfer, is the transfer of DNA from one bacterium to another within the same generation. It often involves DNA recombination, which is a physical process where bits of DNA are mixed together. So you'll have two different sources of the DNA. One will be the bacterial chromosome, the other will be some bit of foreign DNA, and they become one. Prokaryotes will acquire new genes by transformation, which is the process whereby one or more cells die, and DNA fragments from those dead cells are released into the environment. Those fragments are then taken in by bacteria, and through recombination, those fragments and the genes on them are incorporated into the bacterial chromosome of a new cell. We also talked about conjugation, where here plasmids, called fertility factor plasmids, are transferred from one cell to another via sex pilus or a mating bridge. And we now know that these fertility factor plasmids can have other genes on them. Some of them we call dissimilation genes because they allow the metabolism of unusual substances. Some of these plasmids are called R factors because they have lots of antibiotic resistance genes on them. But whatever type of plasmid it is, the hallmark is that there will be genes for making some sort of pilus or mating bridge, which allows the transfer of the plasmids. So antibiotic resistance can be derived by horizontal gene transfer when cells pick up an antibiotic resistance gene on a piece of naked DNA during transformation, or they get a fertility factor plasmid that has resistance genes on it, aka they pick up an R factor. So this is again the end of the first video. In the second video, we'll do a few review questions and we'll talk about two more methods of horizontal gene transfer. I'll see you in that video.